Hello, this is the second lecture in this course Immunology, with the topic being the immune system, with focus on innate and adaptive immunity. The main learning objectives of this lecture are: firstly, to recognize the significance of the immune system in combating infection and disease. Secondly, to know the anatomical, humoral, and cellular components of innate immunity and understand how the innate immune system functions. Thirdly, to know the cells that make up the adaptive immune system and how they function to confer immunity. Fourthly, to be able to explain the types of adaptive immunity. And lastly, to be able to distinguish between the innate and adaptive immune system. The immune system can be defined as a complex network of cells and proteins that defends the body against infection. All cells of the immune system have their origin in the bone marrow, and they include myeloid and lymphoid cells, which differentiate along distinct pathways. The myeloid progenitor cell in the bone marrow gives rise to erythrocytes, platelets, neutrophils, monocytes or macrophages, and the dendritic cells, whereas the lymphoid progenitor cell gives rise to the natural killer cell, B cells, and T cells. For T cell development, the precursor T cell must migrate to the thymus, where they undergo differentiation into two distinct types of T cells, that is the CD4 plus T helper cell and the CD8 plus pre-cytotoxic T cell. There are two types of T helper cells which are being produced in the thymus. These are the T helper 1 cells, which help the CD8 plus pre-cytotoxic cells differentiate into cytotoxic T cells, and the T helper 2 cells, which help B cells differentiate into plasma cells, which secrete antibodies. The main function of the immune system is self versus non-self discrimination. This ability to distinguish between self and non-self is necessary to protect the organism from invading pathogens and to eliminate modified or altered cells. This is so because the immune system recognizes normal cells of the human body as self and modified or altered cells or invading pathogens as non-self. The immune system is made up of two parts: the innate immune system and the adaptive immune system. The innate immune system is the body's first line of defense against pathogens entering the body. It is present from birth and responds in the same way to all pathogens and foreign substances. That is, it is a non-specific type of immune response. The primary function of innate immunity is the recruitment of immune cells to sites of infection and inflammation through the production of cytokines. The innate immune responses can be divided into three types of barriers, which are the anatomical barriers to infection, the humoral barriers to infection, and the cellular barriers to infection. The anatomical barrier is further divided into the mechanical, chemical, and biological factors. Now, let's take a look at some mechanical factors of the anatomical barrier. Firstly, we have the skin, which happens to act as a first line of defense against invading organisms. The epithelial surfaces form a physical barrier that is very impermeable to most infectious agents. Secondly, the flushing action of tears and saliva helps prevent infection of the eyes and mouth respectively. Also, the trapping effect of mucus that lines the respiratory and gastrointestinal tract help protect the lungs and the digestive system from infection. Some examples of the actions of chemical factors of the anatomical barrier are as follows. Fatty acid in sweat inhibits the growth of bacteria. Secondly, lysozyme and phospholipase found in tears, saliva, and nasal secretions can break down the cell wall of bacteria and destabilize bacterial membranes. Thirdly, the low pH of sweat and gastric secretions prevent growth of bacteria. Also, defensins found in the lung and gastrointestinal tract have antimicrobial activity. An example of biological factor of the anatomical barrier are the normal flora of the skin and gastrointestinal tract, which can prevent colonization by pathogenic bacteria by secreting toxic substances or by competing with pathogenic bacteria for nutrients or attachment to cell surfaces. Let's move on to the humoral barriers of the innate immune system. When there is damage to tissues, the anatomical barriers are breached and infection occurs. Once infectious agents have penetrated tissues, another innate defense mechanism comes into play, namely acute inflammation. Humoral factors play an important role in inflammation, which is characterized by swelling and the recruitment of phagocytic cells. These humoral factors are found in serum, or they are formed at the site of infection. Let's take a look at some of the components that make up the humoral barrier. Firstly, we have the complement system. The complement system is a major humoral non-specific defense mechanism. Once activated, complement can lead to increased vascular permeability, recruitment of phagocytic cells, and lysis and opsonization of bacteria. Secondly, we have interferons. Interferons are proteins that can limit virus replication in cells. We also have the coagulation system as part of the humoral barrier. Depending on the severity of the tissue damage, the coagulation system may or may not be activated. Some products of the coagulation system are directly antimicrobial. For example, beta-lysine, a protein produced by platelets during coagulation, can lyse many gram-positive bacteria by acting as a cationic detergent. Fourthly, we have the proteins lactoferrin and transferrin. By binding iron, an essential nutrient for bacteria, these proteins limit bacterial growth. Lastly, we have the interleukin-1, which induces fever and the production of acute phase proteins, some of which are antimicrobial because they can opsonize bacteria. Let's move on to the cellular barriers of the innate immune system. Part of the inflammatory response is the recruitment of polymorphonuclear cells, eosinophils, and macrophages to the site of infection. These cells are the main line of defense of the innate immune system. Neutrophils are recruited to the site of infection, where they phagocytose invading organisms and kill them intracellularly. The second components are the eosinophils, which have proteins in granules that are effective in killing certain parasites. Another component of the cellular barrier of the innate immune system are the natural killer and lymphoid activated killer cells. Natural killer and lymphoid activated killer cells can non-specifically kill virus-infected and tumor cells. Destruction of infected cells is achieved through the release of perforins and granzymes from natural killer cell granules, which induce apoptosis. These cells are not part of the inflammatory response, but they are important in non-specific immunity to viral infections and tumor surveillance. Lastly, we have the macrophages. Tissue macrophages are newly recruited monocytes, which differentiate into macrophages, also function in phagocytosis and intracellular killing of microorganisms. In addition, macrophages are capable of extracellular killing of infected or altered self-target cells. Furthermore, macrophages contribute to tissue repair and act as antigen-presenting cells, which are required for the induction of specific immune responses. Despite the multiple layers of the innate immune system, some pathogens may evade the innate defense. On-call invertebrates is the adaptive immune system, which counters infection with a specific tailor-made response to the attacking pathogen. The adaptive immune system, also referred as the acquired immune system, is a subsystem of the immune system that is composed of specialized systemic cells and processes that eliminate pathogens or prevent their growth. Adaptive immunity develops when innate immunity is ineffective in eliminating infectious agents and the infection is established. The adaptive immune system specifically targets the pathogen causing the infection. But to do that, it first needs to identify the pathogen. This means that it is slower to respond than the innate immune system, but when it does, it is more accurate. It also has the advantage of being able to remember pathogens, so the next time a known pathogen is encountered, the adaptive immune system can respond faster. This memory is also the reason why there are some illnesses you can only get once in your life, because afterwards, your body becomes immune to them. The adaptive immune response can be divided into the cell-mediated immune response, which is characterized by T cells, which are activated through the action of antigen-presenting cells, and humoral immune response, which is characterized by B cells, which differentiate into plasma cells to produce antibodies. Let's take a look at the cell-mediated immune response. T cells are produced in the bone marrow and then move to the thymus through the bloodstream where they mature. These cells express a unique antigen binding receptor on their membrane, known as the T cell receptor, and require the action of antigen presenting cells to recognize a specific antigen. The surfaces of antigen-presenting cells express cell surface proteins known as the major histocompatibility complex. Major histocompatibility complexes are classified as either class 1, which are found on all nucleated cells, or class 2, which are found on only certain cells of the immune system, including macrophages, dendritic cells, and B cells. The class 1 major histocompatibility complex molecules present endogenous peptides, while class 2 molecules present exogenous peptides. The major histocompatibility complex protein displays fragments of antigens when a cell is infected with a pathogen or has phagocytosed foreign proteins. T cells are activated when they encounter an antigen-presenting cell that has digested an antigen and is displaying antigen fragments bound to its major histocompatibility complex molecules. The major histocompatibility complex antigen complex activates the T cell receptor, and the T cell secretes cytokines which further control the immune response. This antigen presentation process stimulates T cells to differentiate into either cytotoxic T cells or T helper cells. Cytotoxic T cells are primarily involved in the destruction of cells infected by foreign agents. They are activated by the interaction of their T cell receptor with peptide-bound major histocompatibility complex class I molecules. Clonal expansion of cytotoxic T cells produce effector cells, which release perforin and gramzyme, which are proteins that causes lysis of target cells, and granulysin, a substance that induces apoptosis of target cells. Upon defeating the infection, most effector cells die and are cleared by phagocytes. However, a few of these cells are retained as memory cells so that future exposure to the same antigen can quickly be handled. T helper cells play an important role in establishing and maximizing the immune response. These cells have no cytotoxic or phagocytic activity and cannot kill infected cells or clear pathogens. However, they mediate the immune response by directing other cells to perform these tasks. T helper cells are activated through T cell receptor recognition of antigen bound to class II major histocompatibility complex molecules. Once activated, T helper cells release cytokines that influence the activity of many cell types, including the antigen-presenting cells that activate them. Two types of T helper cell responses can be induced by an antigen-presenting cell: T helper 1 or T helper 2 response. The T helper 1 response is characterized by the production of interferon gamma, which activates the bactericidal activities of macrophages and other cytokines that induce B cells to make opsonizing and neutralizing antibodies. Meanwhile, the T helper 2 response is characterized by the release of cytokines, which are involved in the activation and or recruitment of immunoglobulin E antibody, producing B cells, mast cells, and eosinophils. Like cytotoxic T cells, most T helper cells will die upon the end of an infection, with a few remaining as T helper memory cells. A third type of T cell, known as the regulatory T cell, also plays a role in the immune response. T regulatory cells limit and suppress the immune system and thereby may function to control aberrant immune responses to self-antigens and the development of autoimmune disease. Let's move on to the humoral immune response, which is mediated by B cells. B cells, which are responsible for humoral immune response, are produced in the bone marrow and undergo maturation there. Following maturation, they leave the marrow expressing a unique antigen binding receptor on their membrane. Unlike T cells, B cells can recognize free antigen directly without the need for antigen-presenting cells. The principal function of B cells is the production of antibodies against foreign antigens. When activated by foreign antigens, B cells undergo proliferation and differentiate into antibody-secreting plasma cells or memory B cells. Memory B cells are long-lived survivors of past infection and continue to express antigen-binding receptors. These cells can be called upon to respond quickly and eliminate an antigen upon re-exposure. Plasma cells, on the other hand, do not express antigen-binding receptors. These are short-lived cells that undergo apoptosis when the inciting agent that induced the immune response is eliminated. Humoral immunity is mediated by B-cell antibody production. The antibody production pathway begins when the B cell's antigen-binding receptor recognizes and binds to antigen in its native form. This in turn attracts the assistance of T helper cells, which secrete cytokines that help the B cell multiply and mature into antibody-secreting plasma cells. The secreted antibodies bind to antigens on the surface of pathogens, flagging them for destruction through pathogen and toxin neutralization, classical complement activation, opsonin promotion of phagocytosis, and pathogen elimination. Upon elimination of the pathogen, the antigen-antibody complexes are cleared by the complement cascade. 5 types of antibodies are produced by B cells, which are immunoglobulin A, immunoglobulin D, immunoglobulin E, immunoglobulin G, and immunoglobulin M. Each of these antibodies have differing biological functions and recognize and neutralize specific pathogens. Adaptive immunity is attained through either passive or active immunization. Passive immunization refers to the transfer of active humoral immunity in the form of ready-made antibodies from one individual to another. It can occur naturally by transplacental transfer of maternal antibodies to the developing fetus, or it can be induced artificially by injecting a recipient with exogenous antibodies targeted to a specific pathogen or toxin. Active immunization refers to the production of antibodies against a specific agent after exposure to the antigen. It can be acquired through either natural infection with a microbe or through administration of a vaccine that can consist of attenuated pathogens or inactivated organisms. Differences between innate and adaptive immunity. The first characteristic we are going to be taking a look at is the response time. The response time of adaptive immunity occurs over days to weeks, whereas that of innate immunity occurs rapidly from minutes to hours. State at birth. Adaptive immunity is acquired during the lifetime of an individual, whereas innate immunity is present from birth. Immunological memory. Adaptive immunity confers immunological memory, whereas innate immunity does not confer immunological memory. Specificity. The response of adaptive immunity is specific against a particular antigen, whereas that of innate immunity is the same against all invading pathogens, that is, it is non-specific. Subsequent exposure. With adaptive immunity, immune response improves with each subsequent exposure, whereas with innate immunity, the immune response does not change on repeated exposure. Order of defense. Innate immunity is the first line of defense of the immune system, whereas adaptive immunity is the action against pathogens that are able to overcome innate immune defenses. Response-directed sites. Adaptive immunity is directed towards specific epitopes, whereas innate immunity is directed towards types of molecules. Adaptive immunity is found only in vertebrates, whereas innate immunity is found in nearly all forms of life. We've come to the end of this lecture. Before you leave, don't forget to hit the subscribe button, Give this video a thumbs up, turn on your notification, and follow us on all our social media handles.